Hi, today's lecture is going to be on nervous tissue. So your nervous system is going to be everything. This is what's going to control everything inside of your body. The nervous system does work along with one other system, and that is going to be your endocrine system. That one's going to be very important as well. So both of those working together will control everything that goes on in your body. We will cover the endocrine system more in the second part of anatomy in 142. So looking here at this animal looks very angry, very mad. It's got that crazy look in its eyes. This animal has rabies. Rabies is going to be a virus that is known for attacking the nervous system and the central nervous system of a body. So how this actually works is once you are bitten by a rabid animal, the saliva carries that virus. That virus then will travel through the system into the bloodstream and then eventually make its way to the brain and cause inflammation of the brain. It's usually transmitted, like we said, through a wound, through a bite, and it's from an animal who has already been infected. Now looking here, this is what the virus actually looks like. It's a bullet-shaped virus. It is very contagious, very infectious. Inside of it, you will see that it does have an RNA version of DNA. It's going to have proteins that are going to be found throughout it. It is covered in a cell membrane or an envelope, and you notice that it does have that rod shape to it. Just a minute. Sorry, my screen froze for a minute. Now, in the state of Virginia, where we're located, you'll see that there are many different types of animals who can get rabies. Some of the most common ones that you might see them in are going to be things like skunks, raccoons, foxes. You might see wild cats out, bats, and the possum. Other animals can also get rabies. Horses could get rabies. Dogs, cats could get rabies. It's very common. And so one thing to look for is an unusual aggressive behavior in an animal, and behavior that you've not noticed before. If it's an animal that usually does not approach humans, be careful with that as well, because rabid animals usually tend to approach humans and they seem to be very aggressive, very angry, and they do try to snap at you. Now, the nervous system is going to be divided up into two parts. You're going to have the central nervous system and you're going to have the peripheral nervous system. So, looking here, the central nervous system is mainly going to be made up of the brain and spinal cord. So, next week's lecture will be on the brain and spinal cord. This week's lecture, we're talking about nervous tissue. But in order to talk about nervous tissue, we kind of put it all together. So, everything in purple that you see in this box is going to be the peripheral nervous system. The peripheral nervous system is going to be all of the nervous tissue outside of the central nervous system. So if the central nervous system is going to be the brain and spinal cord, the peripheral nervous system is going to be the nerves that are coming off and all the tissue that has to regulate everything that's going on in the body. So we'll start over here with the somatic nervous system, often referred to as the SNS. So with the somatic nervous system, you've got the somatic and special sensory receptors and somatic sensory neurons. So those are going to pick up on information throughout the body. That's going to send it to the brain. The brain then it sends it through the spinal cord to the brain. The brain decides on a response, and it will send that response out through a motor response. So that's going to be a somatic motor response. And so that sends it to the skeletal muscles, which will then respond. The next is going to be what's called the autonomic nervous system, or ANS. So the autonomic nervous system has sensory receptors and autonomic sensory neurons, which are going to pick up on information in the sympathetic and parasympathetic nervous system. So it picks up that information, sends it to the brain, and the brain makes a decision of what needs to be done. It's going to send out that response through the autonomic motor neurons, which are going to be your involuntary muscles and involuntary reactions. And so if it is sympathetic, then that's going to be a fight or flight response that's going to react. So it can send out adrenaline to different areas that are going to help to respond very quickly to a dangerous situation. If it's part of the parasympathetic autonomic nervous system or division that it's sending it out. That's going to be your body when it's in its normal resting phase or when you're going about your daily routine activities. The last is going to be the enteric nervous system or the ENS. So there, with those enteric motor neurons, those are going to be involuntary and it's going to be processed at the enteric plexus. And so that's going to be things such as your smooth muscle, your glands, your endocrine glands, and your GI tract. So it's constantly surveying and picking up information in that area. It might send it to the brain to be processed to see if anything needs to be done. It will then send a response out, usually through the autonomic nervous system, which will then come in and cause a change. So the nervous system cellular structure is going to be made up of what's known as nervous tissue. Nervous tissue could be either the neurons, which are going to be the main conducting cells that will conduct your nerve impulses. You also have the neuroglial cells. These neuroglial cells are going to be the support cells. So if you're looking at this picture, the main portion of that larger cell that you see in darker purpley pink, that is going to be the neuron. That neuron has extensions coming out of it. And so with those extensions, let's see if I can get my pen here, we'll do light blue. It's going to have extensions. There we go. These extensions here are going to be your dendrites. So with these dendrites that you see coming out here, those are going to pick up on the information. It's going to travel in through the cell body where it then turns into a synaptic message or nerve impulse. And then it travels out this section here, which is going to be referred to as the axon. Now, surrounding that cell, which is going to help to protect it, help to keep these cells in the correct space, kind of give it a structure, make sure it's nourished and has everything that it needs. Those little cells are going to be called neuroglial cells, and you'll have many, many more neuroglial cells than you actually have of the nerve cells. So looking here, this is just going to show you some different types of cells that you're going to find inside of the nervous system. So this is different types of nervous tissue. So up top, you'll see some epidermal cells. These epidermal cells are going to be responsible for being in the central nervous system. They create things such as cerebral spinal fluid, which you're going to see throughout the notes as CNS or CNF. You'll see some regular neuron cells. These regular neuron cells are going to be responsible for conducting nerve impulses and picking up on information and monitoring the system. You'll notice that with these, you'll also have some astrocytes. These astrocytes are going to be very important in holding the shape of the nervous tissue, keeping everything kind of there and in place. And they're also going to be responsible for creating a blood-brain barrier between the blood cells, the arteries, and the veins. You'll see how they attach into these blood vessels here, but they're going to have tight junctions and not allow that the different types of substances that are going to be in the blood to get out of the blood into the brain. You'll also notice that coming off of the neurons, you're going to have axons, and those axons will be surrounded by a fatty tissue. So that fatty tissue is going to be referred to as a myelin sheath. So you see that there in yellow. Now you're going to have two different types of cells that create this tissue. They're going to be, if it's in the central nervous system, they're going to be referred to as the oligodendrocytes. And so they'll wrap around those axons and they'll insulate them. If it's going to be found in the peripheral nervous system, these are going to be Schwann cells. And those Schwann cells will make that myelinated sheath that goes around the axons. So here's simply going to be a picture of nervous tissue. And so once you get used to looking at nervous tissue underneath the microscope, you'll be able to pick out different things such as the neurons. You'll be able to pick out axons. You'll be able to pick out neuroglial cells. These here are going to be artist renditions. So looking at these, you can see the main cell bodies, and then you can see all the different dendrites that are coming off of these cell bodies. And it shows you how complex these cells can be in the nervous system. This is going to be an electron micrograph picture of a neuron, and so this is done with a computer image it goes in and it actually shows you what it looks like inside of that tissue. Now, these are always done in black and white unless they add what's known as a false coloring to it. And so here you see that false coloring added to the main neurons, and you see all of the neuroglial tissue that's surrounding it, kind of keeping it in place and taking care of it. This is showing you a motor neuron with neuromuscular junctions attached to it. So you see the axons coming down, you'll see the synaptic clefts, and you'll see how they attach to these muscle cells here. So this is the muscle cell, and these are going to be the neuromuscular junctions. The whole motor neuron or motor unit is going to be this that comes down off of it. So once an action potential, which is a nerve signal, gets sent down through the axon, it's going to be an all or nothing response. So it can bifurcate, it can split off and go into different directions, attached to different muscle fibers. As it goes down and splits off, every one of those that it's attached to will contract and will respond. Here you have a neuromuscular junction under a transmission electron microscope. So it shows you that you're going to have like the synaptic cleft through this area here. You have that presynaptic membrane through here. You're going to have the postsynaptic membrane with some of those invaginations. That's where that sarcolemma is going to go and actually bend in on itself. That creates things such as T-tubules in some spots. You'll see that the arrows pointing at these vesicles here, they're going to be filled with a neurotransmitter. So if this was attached to a muscle cell, these would possibly be filled with acetylcholine. And this is going to be a light micrograph picture showing a motor unit attached to skeletal muscle cells. And this is going to show the green fluorescent protein, or GFP, that's going to be found naturally in animals. They have learned to isolate this protein and take this protein out and be able to insert it into other things or other animals. Once it's applied to a black light or a UV light, then it glows. Under normal conditions, under regular light, you would not see it glowing. So they've been able to isolate this protein and insert it into things so they can make them glow. All right, so here's some animals that they have inserted this GFP protein into. And you can see how under UV light or under a black light that they glow. So we have some cute little monkeys. We have a cat, we have a rat, and we have a rabbit here. Here they took that protein and they inserted it into one neuron. And so you can see the cell body, which is that main portion of that neuron. And then you can see the different dendrites coming out and how they branch out and how intricate they are. And this here shows another picture where they went in and inserted this GFP into different neurons. And here's going to be some different proteins that they found that will show up in different colors and that they've been able to isolate and insert into different living things. One animal that you might have noticed that carries this different protein are going to be these fish here. Now, these fish don't naturally have these different colors. What they do is they take these different proteins, they inject them into the fish, and then usually there's going to be some type of UV light or black light that's going to be in with that tank, and it's going to cause the fish to look like they glow. So they will exhibit this color. Normally, they do not have these colors in them. Here they've went in and they've inserted the protein into each one of the neurons so you can see how intertwined and intricate of a system that they make. And same thing here. Here they went in and they inserted the different colored proteins into different neuromuscular junctions so you could see where they lead and where they're connected at. And all of these, they almost make beautiful works of art. Here you can see where they took a neuron and they cut into it, and you can see the different dendrites that are coming out of it and the different signals going down the axon here. And so here you see all the different dendrites that could be attached to the cell body, and they are going to go out and attach in and connect to other neurons. All right, so neurons are going to be excitatory cells that can change their voltage and become excited. there's different types of neurons. You can have sensory neurons, which are going to collect and send information to the brain and spinal cord about the inside or outside of the body. You have interneurons. These are going to be found in the brain or in the spinal cord, and they're going to connect neurons and process information and send messages direct motor neurons, which are going to do the responses. And of course, the motor neurons, like I said, are going to carry those nerve impulses from the brain to the spinal cord, and then from the spinal cord to the muscle or to the glands, and that's going to be the response from the brain. Stimulus is going to be a change that's going to initiate an action potential. So a type of stimulus that you may have may be that you touched a hot stove. So that stimulus is going to be the hot. It's going to travel to the spinal cord, and some things, such as something that's very hot or very sharp, that decision needs to be made right away. And so you're going to have the neurons that are going to be in the spinal cord that can make reflex decisions right away. And so they'll send out that action potential to cause that movement. So the action potential is going to be the nerve impulse, and that travels along the axon very, very rapidly. And so here, you're going to see a visual. They see a glass of water. That input gets sent to the brain. The brain processes it and thinks, hey, let's pick up that glass of water. We're kind of thirsty. So that's going to be that integration or the interneuron. And then it's going to send a message out through the motor neuron as a motor output. So it's going to activate those muscles to pick up that glass of water, and then you can have a sip. This picture here is going to be the picture of an average neuron. It's going to contain all the basic things that a neuron has. So the main portion of the neuron will be the cell body. So that's going to be this section right through here. So that's your cell body. Inside the cell body, it's going to have things like mitochondria, ribosomes, smooth endoplasmic reticulum, rough endoplasmic reticulum. All of your different organelles that you will find in a regular cell will be found here. On the edges, the ones that are picking up information, all of these are going to be referred to as dendrites. So you have your dendrites out through here. So they pick up information, it travels into the cell body, and midway through, as it makes its way to the portion of the cell body that starts to come in on itself, that's going to be called the axon hillock. Around this area is where that stimuli is going to be changed into a nerve impulse or an action potential. Same thing. That action potential will then travel down the axon. So it travels down the axon there. Now noticing there, you're going to see that you have these different cells that are going to be wrapped around that axon. Those are the cells that I told you are going to insulate the axon. That insulation is going to be very, very important. So it's going to travel down the axon through these insulated cells, which are going to help to kind of speed up and send that signal along very quickly. Now, if it's going to be found in the peripheral nervous system, that will be your Schwann cells. And if it's found in the central nervous system, that's going to be your oligodendrocytes. So that information will travel down until it makes its way to the end of the axon. Once it makes its way to the end of the axon, that's where you're going to find axon terminals and synaptic bulbs at the end. That's going to connect to another neuron and keep sending that information through, or it could connect to something like a gland or to a muscle cell. So going back to our, going back to rabies. So what happens? The virus is going to enter the person through an animal bite. From there, that virus replicates in the muscle at the site of the bite. Then the virus infects the nerves in the peripheral nervous system, and it moves through what's known as a retrograde transport. That virus replicates itself in the dorsal root ganglion and the spinal cord, and it then is going to travel up the spinal cord to the brain. From there, the brain becomes infected. That virus then travels from the brain through nerves to other tissues in the body, such as the eyes, the kidneys, the salivary glands. That's why people and animals with rabies will start to salivate quite a bit. They often say that if you see a dog or an animal foaming at the mouth, that's because those salivary glands have been activated from this rabies virus. And so that's a good indication that that animal could have rabies. All right, So looking here, this is showing you the average nerve cell or neuron, and you notice that it says it has about 10,000 Schwann cells in 1 meter of an axon. So that's how small this axon can be and this neuron can be, and how many of those cells are going to be wrapped around that axon to help to insulate that nervous system, that neuron as it's traveling. So with this neuron, notice how it shows you the impulse direction. That impulse direction is very important. The impulse is always going to travel away from the cell body. So it travels not away from the cell body, it travels from the dendrites through the cell body. But once it reaches the axon, it travels down the axon away from the cell body until it makes it to the N terminals. Then it might connect to another another neuron, where it's going to come in through its dendrites, travel through the cell body, and then down its axons. All right, so here you will see with this one, you see the dendrite, you see the cell body, and then you see that axon hillock. That's going to be that region of the axon as it starts to come in and get smaller, and it's going to be unmyelinated. Then you will hit a myelinated region, and then you hit another region that's not myelinated. Once you start to hit those regions after the axon hillock that are not myelinated, those are going to be referred to as the nodes of Ranvier. And those are going to be important in just a minute when we talk about how the nerve action potential actually travels down the axon. Notice when you're looking at it in the picture that's enlarged, how that myelin looks like it has circles going around it. That's because that cell will continuously wrap around the axon many, many times, creating a fatty layer that's going to be many layers thick. And so the cell portion is going to actually be on the outer portion with that nucleus and its organelles. So in this case, this one's showing you a Schwann cell. So you know that this neuron will be found in the peripheral nervous system. This is what it actually looks like underneath a cross-section of an electron micrograph. And so you can see that myelin sheath, you can see how it's many layers thick with that adipose tissue that's wrapped around it. And then of course you have your outer color in that area, which is going to contain the actual cell. So myelin sheath is going to be produced by Schwann cells in the peripheral nervous system, or it will be produced by the oligodendrocytes in the central nervous system. And these are going to be what start to develop around the axon. So here you see a Schwann cell starting to develop around this axon. It's going to start to wrap its extensions, cytoplasmic extensions around. After it starts to wrap it around several times, then it's going to change over into that myelin or into that fatty substance. Now, as you're learning things, as your axons are connecting, you are going to start to make these connections. Dendrites will connect to axons, and then axons will travel further down, connect into more dendrites. As they do this, as you're learning, these connections are being made. So over time, this myelin is going to start to produce. So you'll have either Schwann cells, if it's a muscle that is starting to learn a memory, or when it goes to the brain, it's going to be the oligodendrocytes, which are creating these myelin connections. These are going to help you to learn and to solidify that information. So this will help with things such as muscle memory, or it actually helps with actual memory in the brain. Now, it does take time for it to do this. This is not going to be something that you will learn 5-10 minutes in class and then you can keep with you. These connections are going to need to be practiced and solidified. So as it's continuing to wrap this myelin around these axons, that's when it starts to stick. That's when it starts to solidify. So that's why it's so important to actually go in and study a little bit each night. Now, as you're a child and you're starting to learn to walk, these muscles and the axons that are attached to it, they have to myelinate. So it's going to take a while for those muscle movements to become smooth and to become executed at an almost expert way. Here's a video on multiple sclerosis, which you can watch on the PowerPoint that I have in Canvas. here, this is going to show you, well, this is going to show you the different structural classifications of neurons. So the first one's going to be referred to as an axonic neuron. This axonic neurons is going to have two processes, and they will all be in those dendrite sections. And so the axons are not going to be obvious to you here. The next, you have your bipolar neuron. And with this type of neuron, they're going to have two processes that are going to be separated by a cell body, which you see there in the center. So on one side, you'll have your dendrites. On the other side, you have your axons. The other one is going to be referred to as a unipolar neuron. And this one's going to have a single elongated process that comes out of the cell body, but then it's going to split. And so as it splits, some of those will split off and make axons on either side that will have dendrites in that initial segment. And then you have your multipolar neurons. These neurons are going to have more than two processes, and there's going to be a single axon and multiple dendrites coming out. So some neurons are going to be named by the scientists that found them, such as like the Schwann cell that wraps around neurons. This one here is called a Purkinje's cell. This is going to be found in the heart, so it's named after the scientists that found them. And then sometimes they're going to be named by shape. So this is a pyramidal cell because it's shaped like a pyramid in the cell body formation. Now, neuroglial cells we talked a little bit about. Their function is to support and provide structural framework for the neurons. They also will fill up a lot of space. With these neuroglial cells, neuro means nerve, glial is going to be glue. They also can produce some myelin insulation that's going to be found in the nervous tissue. And they can carry out phagocytosis. So if they come in contact with microorganisms that are going to be found in the neural tissue, such as viruses or bacteria, they can engulf them and destroy them using enzymes in the lysosomes. Or they can take cellular debris from old worn-out cells that have kind of started to break down, or neurons that have become damaged, and they can break those down and get rid of those. Astrocytes are going to be a form of structural support that these cells will make. They actually make what I call that blood-brain barrier, and so they can form a barrier between the brain and the capillaries. This is going to be important because it will allow things such as the plasma through, but it does keep other things out. So the cells can actually make it through, and chemicals can't make it through for the most part. So if, let's say, you're taking a chemotherapy drug, it will not help with brain cancer in this case because it can't make it through this blood-brain barrier. But that's going to be very important in protecting the brain from different types of poisons and chemicals. Next, you have your microglial cells. These are going to be cells that are going to help with support, and they also help carry out phagocytosis for anything that does not belong in that tissue. Next, we have our ependymal cells. These are going to line the ventricles and the central canal of the brain and spinal cord. These are going to be very important in making cerebral spinal fluid, which you may see abbreviated as CSF throughout your notes. These are going to be found in the central nervous system. Next, we have the oligodendrocytes. These provide the myelin for neurons in the brain and spinal cord, so this is going to be another central nervous system cell. And here you're looking at the neuroglial cells of the PNS, or peripheral nervous system. And so with these, you'll notice that you have satellite cells that start to form around. These satellite cells might then start to work their way around that neuron cell body and in that ganglion. And coming back for a minute, there's something, let me see. No. Here, notice the Schwann cell, how it's developing and how it's wrapping around in with that. And so here I forgot to tell you about MS, or multiple sclerosis. This is where these Schwann cells become damaged. And if a Schwann cell becomes damaged and you no longer have this myelin around these axons, then those messages that are traveling through the axons start to slow down so they don't travel as fast. Eventually, the destruction of the Schwann cell or the oligodendrocytes will make those axons completely unmyelinated, and so that can actually cut off that message or the synapse that's traveling down that axon completely, so it never makes it. So over time, you start to lose control of nerve or of muscles and of the body in these areas. And this is where we're going to stop for the first part of this video. I will pick up with nerve cell physiology in the next video.